Gene therapy is a revolutionary approach in modern medicine that aims to treat or prevent diseases by modifying a person's genetic material. Unlike traditional treatments that often address only the symptoms, gene therapy targets the root cause at the DNA level. This innovative technique can involve replacing faulty genes, inactivating malfunctioning ones, or introducing new genes to help the body fight or prevent disease. As science advances, gene therapy is becoming a beacon of hope for conditions once thought untreatable, such as certain genetic disorders, cancers, and viral infections. Let's explore how this cutting-edge science is changing lives. Genes are the fundamental units of heredity, carrying instructions that determine everything from eye color to how our bodies function. They are made up of DNA, a complex molecule that encodes the information necessary for building and maintaining our bodies. When genes work properly, they help keep us healthy. However, mutations or errors in our genetic code can lead to a variety of diseases. Understanding how genes operate is crucial for grasping the potential and challenges of gene therapy. This foundational knowledge sets the stage for the remarkable possibilities that gene therapy offers. Gene therapy works by delivering new or modified genes into a patient's cells. This can be done using various methods, such as viral vectors, which are engineered viruses that carry healthy genes into cells, or non-viral techniques like direct injection of genetic material. The goal is to correct or compensate for defective genes responsible for disease development. Once inside the body, these new genes can start producing the necessary proteins or enzymes that the patient's body lacks. This process can lead to significant improvements in health and, in some cases, offer a potential cure. Gene therapy can be classified into two main types: somatic and germline. Somatic gene therapy targets non-reproductive cells, meaning changes affect only the treated individual and are not passed to offspring. Germline gene therapy, on the other hand, involves altering genes in eggs, sperm, or embryos, making changes heritable. Currently, most gene therapy research and treatments focus on somatic cells due to ethical and safety concerns surrounding germline modification. Understanding these types helps clarify the scope and implications of gene therapy in medicine today. Journey of gene therapy began in the 1970s, with scientists envisioning the possibility of correcting genetic defects at their source. The first approved gene therapy trial took place in 1990, treating a young girl with a rare immune disorder. Since then, the field has experienced both breakthroughs and setbacks, including early challenges with safety and effectiveness. Over the decades, advances in molecular biology and technology have propelled gene therapy forward, leading to successful treatments for certain inherited diseases and cancers. This rich history highlights the persistence and innovation driving the field. Gene therapy has reached several significant milestones over the years. Notable achievements include the first successful treatment of severe combined immunodeficiency (SCID), advancements in treating inherited blindness, and the approval of gene therapies for certain blood disorders. Each milestone represents years of research, collaboration, and perseverance. These successes have paved the way for more therapies to enter clinical trials, offering hope to patients with previously untreatable conditions. The journey continues as researchers strive to expand the reach and effectiveness of gene therapy. Today, gene therapy is being used to treat a growing list of diseases. These include rare genetic disorders like spinal muscular atrophy and hemophilia, as well as certain types of cancer. In some cases, gene therapy offers the only available treatment option, providing life-changing results. The therapies are often tailored to individual patients, reflecting the personalized nature of this approach. As more therapies gain approval, the impact of gene therapy on modern medicine continues to expand, bringing new hope to patients and families worldwide. One of the most promising uses of gene therapy is in treating genetic disorders caused by single gene mutations. Conditions like cystic fibrosis, sickle cell anemia, and Duchenne muscular dystrophy are being targeted with gene therapies designed to correct or replace the faulty gene. These treatments can dramatically improve quality of life and, in some cases, offer a potential cure. Ongoing research continues to expand the list of treatable genetic diseases, bringing hope to patients who previously had limited options. Gene therapy is making significant strides in cancer treatment. By modifying immune cells to better recognize and attack cancer cells, therapies like CAR T-cell therapy have achieved remarkable results in certain blood cancers. Other approaches involve introducing genes that make cancer cells more susceptible to existing treatments or that directly kill tumor cells. These innovative strategies are transforming the landscape of cancer care, offering new hope to patients who have exhausted traditional options. Gene therapy is also being explored as a way to combat viral infections, such as HIV and hepatitis. By introducing genes that help the body resist or fight off viruses, researchers hope to develop long-lasting treatments or even cures. Some experimental therapies aim to make immune cells resistant to infection, while others seek to eliminate the virus from the body entirely. These approaches are still in development, but they represent a promising frontier in the fight against infectious diseases. Delivering genes into the body is a complex process. Scientists use vectors, which are carriers that transport genetic material into cells. The most common vectors are viruses that have been modified to be safe and effective. Non-viral methods such as injecting naked DNA or using nanoparticles are also being developed. The choice of delivery method depends on the disease, the target cells, and safety consideration. Effective delivery is crucial for the success of gene therapy, and ongoing research aims to improve these techniques. Viral vectors are engineered viruses used to deliver therapeutic genes into cells. These viruses are modified so they cannot cause disease, but they retain their ability to enter cells and deliver genetic material. Common types include adenoviruses, lentiviruses, and adeno-associated viruses. Each type has unique advantages and challenges, such as how long the gene stays active and which cells it can target. Viral vectors have been central to many successful gene therapy treatments, but researchers continue to refine them for greater safety and efficiency. Viral methods for delivering genes are gaining attention due to their potential safety advantages. Techniques include direct injection of DNA, using nanoparticles, or employing physical methods like electroporation. These approaches avoid the risks associated with viruses such as immune reactions, but often face challenges in efficiency and targeting. Researchers are working to improve these methods, aiming to make gene therapy safer and more widely applicable. As technology advances, non-viral delivery could play a larger role in future treatments. Gene therapy is at the forefront of personalized medicine, tailoring treatments to an individual's unique genetic makeup. By analyzing a patient's DNA, doctors can design therapies that specifically target the underlying cause of disease. This approach increases the chances of success and reduces the risk of side effects. Personalized gene therapies are already making a difference in rare diseases and cancer, and their use is expected to grow as genetic testing becomes more accessible and affordable. Gene therapy raises important ethical questions. Issues include the potential for unintended genetic changes, the use of germline therapy, and concerns about access and equity. There are also debates about the possibility of designer bodies and the long-term effects of altering human DNA. Ethical guidelines and regulations are in place to ensure that gene therapy is used responsibly and safely. Ongoing dialogue among scientists, ethicists, and the public is essential as the field continues to evolve. Safety is a top priority in gene therapy research. While many treatments have shown promising results, there can be risks such as immune reactions, unintended genetic changes, or insertional mutagenesis. Researchers carefully monitor patients during and after treatment to detect any adverse effects. Advances in technology and a better understanding of genetics are helping to minimize these risks. As gene therapy becomes more common, ongoing vigilance will be crucial to ensure patient safety. Gene therapy treatments must undergo rigorous testing and regulatory review before they can be approved for use. Agencies like the FDA and EMA evaluate the safety, effectiveness, and quality of new therapies. Clinical trials are conducted in phases, starting with small groups of patients and expanding as more data is collected. Only after demonstrating clear benefits and manageable risks can a gene therapy receive approval. This careful process helps protect patients and ensures that new treatments meet high standards. Gene therapy can be expensive, often costing hundreds of thousands or even millions of dollars per treatment. These high costs reflect the complexity of developing, manufacturing, and delivering these therapies. Accessibility is a major concern, as not all patients can afford or access these treatments. Efforts are underway to reduce costs and expand access, including insurance coverage, government programs, and innovative payment models. Making gene therapy available to more people remains a key challenge for the field. Ongoing research is the backbone of gene therapy's progress. Scientists around the world are working to develop new techniques, improve safety, and expand the range of treatable diseases. Clinical trials are essential for testing new therapies and gathering data on their effectiveness. Collaboration between universities, hospitals, and biotech companies accelerates innovation. As research continues, we can expect even more breakthroughs in the years ahead, bringing gene therapy closer to mainstream medicine. Gene editing technologies like CRISPR-Cas9 have revolutionized the field of gene therapy. These tools allow scientists to precisely modify DNA, correcting mutations or adding new genetic material. CRISPR is faster, cheaper, and more accurate than previous methods, making it a game-changer for research and treatment. While still in the early stages of clinical use, gene editing holds enormous promise for treating a wide range of diseases and advancing our understanding of genetics. CRISPR technology is already being used in experimental gene therapies. For example, researchers are using CRISPR to correct genetic mutations in blood cells to treat sickle cell anemia and beta thalassemia. Early results are promising, with some patients experiencing significant improvements. CRISPR's precision reduces the risk of unintended changes, but careful monitoring is still necessary. As more clinical trials are conducted, CRISPR-based therapies could become a standard part of gene therapy in the future. Despite its promise, gene therapy faces several challenges. Delivering genes to the right cells, avoiding immune reactions, and ensuring long-term effectiveness are ongoing hurdles. Manufacturing therapies at scale and ensuring consistent quality are also complex tasks. Additionally, ethical, regulatory, and financial issues must be addressed. Overcoming these challenges requires continued research, collaboration, and innovation. The field is advancing rapidly, but there is still much work to be done. There are inspiring stories of patients whose lives have been transformed by gene therapy. Children with rare immune disorders, adults with inherited blindness, and cancer patients have all benefited from these groundbreaking treatments. These success stories highlight the real-world impact of gene therapy and the hope it brings to families. While not every patient responds the same way, each success fuels further research and inspires others to keep pushing the boundaries of what's possible. Patients' gene therapy can mean a new lease on life. Many describe the experience as life-changing, offering hope where there was once none. However, the journey can also be challenging, with uncertainty about outcomes and potential side effects. Support from medical teams, family, and patient communities is essential. Listening to patient voices helps researchers and doctors improve therapies and provide better care. Their stories remind us of the human side of scientific progress. Gene therapy doesn't just affect patients, it impacts entire families. Parents of children with genetic disorders often face years of uncertainty and worry. Successful gene therapy can bring relief, hope, and a brighter future for the whole family. At the same time, families may have concerns about long-term effects and access to treatment. Open communication with healthcare providers and support networks can help families navigate these challenges together. Doctors play a critical role in gene therapy, from diagnosing eligible patients to administering treatments and monitoring outcomes. They work closely with researchers to stay updated on the latest advances and ensure patients receive the best possible care. Education and communication are vital, as patients and families often have many questions about gene therapy. By guiding patients through the process, doctors help turn scientific breakthroughs into real-world solutions. Gene therapy is a collaborative effort involving scientists, doctors, patients, and industry partners. Research teams work across disciplines, combining expertise in genetics, molecular biology, and medicine. Collaboration accelerates progress, allowing new ideas to be tested and therapies to be developed more quickly. Partnerships with biotech companies help bring treatments from the lab to the clinic. This spirit of teamwork is essential for overcoming challenges and achieving breakthrough. Advances in technology have been crucial to the development of gene therapy. High-throughput DNA sequencing, advanced imaging, and powerful computing tools allow scientists to analyze genes and design therapies with unprecedented precision. Automation and robotics help scale up manufacturing, making treatments more widely available. As technology continues to evolve, it will open new possibilities for gene therapy and other forms of personalized medicine. Producing gene therapies is a complex and highly regulated process. It involves growing cells, engineering vectors, and ensuring the final product meets strict quality standards. Manufacturing must be done in specialized facilities to prevent contamination and ensure patient safety. Scaling up production to meet demand is a major challenge, but advances in biotechnology are making it more feasible. Reliable manufacturing is essential for bringing gene therapies to more patients. Clinical trials are the backbone of gene therapy development. These studies test new treatments in volunteers to assess their safety and effectiveness. Trials are conducted in phases, starting with small groups and expanding as more data is collected. Participation is voluntary and patients are closely monitored throughout the process. Clinical trials provide the evidence needed for regulatory approval and help refine therapies for broader use. Gene therapy research and treatments are happening around the world. International collaboration helps share knowledge, resources, and expertise. Some countries are leading the way in clinical trials and approvals, while others are working to build capacity. Global efforts are essential for ensuring that gene therapy benefits as many people as possible, regardless of where they live. The worldwide impact of gene therapy is growing every year. Future of gene therapy is filled with exciting possibilities. Researchers are exploring ways to treat more diseases, improve delivery methods, and make therapies safer and more affordable. Advances in gene editing, artificial intelligence, and biotechnology will drive further innovation. As our understanding of genetics deepens, gene therapy could become a routine part of medical care, transforming how we treat and prevent disease. Some researchers are investigating whether gene therapy could help slow or reverse aspects of aging. By targeting genes involved in cell repair and regeneration, scientists hope to extend healthy lifespan and reduce age-related diseases. While this area is still in its early stages, it holds great promise for the future. Successful gene therapy could play a key role in promoting healthy aging and improving quality of life for older adults. Rare diseases, often caused by single gene mutations, are a major focus for gene therapy. Many of these conditions have no effective treatments, making gene therapy a beacon of hope for patients and families. By correcting or replacing the faulty gene, these therapies can dramatically improve health and quality of life. Success in rare diseases also provides valuable lessons for treating more common conditions in the future. Children with genetic disorders are among the primary beneficiaries of gene therapy. Early intervention can prevent serious complications and improve long-term outcomes. Pediatric gene therapy requires special considerations, including dosing, safety, and long-term monitoring. Success stories in children inspire hope and drive further research. As more therapies become available, the outlook for young patients with genetic diseases continues to improve. Gene therapy has shown remarkable success in treating certain inherited eye diseases. By delivering healthy genes to retinal cells, these therapies can restore vision in patients with conditions like Leber congenital amaurosis. The eye is an ideal target for gene therapy due to its accessibility and immune privilege. These breakthroughs are paving the way for new treatments for blindness and other vision disorders. Blood disorders like hemophilia and sickle cell anemia are among the first diseases to benefit from gene therapy. By introducing functional copies of the faulty gene, these therapies can reduce or eliminate symptoms. Some patients have achieved long-term remission after a single treatment. These successes demonstrate the power of gene therapy to transform lives and offer hope for other blood-related conditions. Muscular disorders like Duchenne muscular dystrophy are being targeted with gene therapies designed to restore muscle function. By delivering healthy copies of the affected gene, these treatments aim to slow disease progression and improve mobility. Early clinical trials have shown encouraging results, with some patients experiencing increased strength and independence. Continued research is needed to refine these therapies and expand their use. Neurological diseases like spinal muscular atrophy and certain forms of epilepsy are being addressed with gene therapy. By delivering genes that support nerve function or correct underlying defects, these therapies can improve symptoms and quality of life. The brain and nervous system present unique challenges for gene delivery, but advances in technology are making progress possible. These breakthroughs offer hope for patients with debilitating neurological conditions. Researchers are exploring gene therapy as a treatment for heart disease. By introducing genes that promote blood vessel growth or repair damaged tissue, these therapies aim to improve heart function and prevent complications. While still in early stages, gene therapy could become an important tool in managing cardiovascular diseases, which are a leading cause of death worldwide. Ongoing trials will determine its effectiveness and safety. Gene therapy is being investigated as a potential cure for diabetes. By modifying genes involved in insulin production or immune regulation, researchers hope to restore normal blood sugar control. Early studies in animals and humans have shown promise, but more research is needed before these therapies become widely available. If successful, gene therapy could revolutionize diabetes care and reduce the burden of this chronic disease. Beyond HIV and hepatitis, gene therapy is being explored for other infectious diseases, including COVID-19. By enhancing the immune system's ability to fight off pathogens, these therapies could provide long-lasting protection. Some approaches involve engineering immune cells to recognize and destroy infected cells. While still experimental, gene therapy could become a powerful tool in the fight against emerging infectious diseases. Public perception of gene therapy is evolving as more success stories emerge. While some people are excited about the possibilities, others have concerns about safety, ethics, and long-term effects. Education and transparent communication are key to building trust and understanding. As gene therapy becomes more common, public attitudes will play an important role in shaping policy and access to treatment. Gene therapy is still a young field, but its potential is immense. Continued research, collaboration, and innovation will be essential for overcoming challenges and expanding access. As more therapies are developed and approved, gene therapy could become a standard part of medical care. The journey is just beginning, and the future holds endless possibilities for improving health and saving lives. Summary: gene therapy represents a major leap forward in medicine, offering hope for patients with a wide range of diseases. From correcting genetic defects to fighting cancer and infectious diseases, the possibilities are expanding every year. While challenges remain, the progress made so far is inspiring. With continued dedication from scientists, doctors, and patients, gene therapy has the potential to transform healthcare and change countless lives for the better.